This is Selma Schimmel in Chicago at the annual ASCO meeting for the group room, and we're very happy to sit with you, Dr. Clifford Hudis, Chief of the Breast Cancer Medicine Division at Memorial Sloan-Kettering Cancer Center in New York City. Hi, Dr. Hudis.
Hi.
Thanks for being here.
It's a pleasure.
I think it would be appropriate to take a moment out, since we're really talking mostly to patients, to acknowledge and thank women with breast cancer who take the step to get involved in a clinical trial, and acknowledge them, because without our involvement, not only do we get breast cancer, but we get involved in the research, and it takes courage and an understanding of how you bring meaning to your own experience.
Well, this year's meeting had a couple of call-outs, I would say, in this regard, which wouldn't necessarily have been obvious to you, but the presidential speech that Michael Link gave was a reflection on his work as a pediatric oncologist. And the pediatric oncologists have something I think important to teach us. Their diseases are all by definition rare. They only succeed through collaboration. And they've spent decades making participation in clinical trials the way the diseases are treated. You only go to standard therapy, so-called standard therapy, off-study treatment when you have no alternative. We as adult oncologists really seem, unfortunately, to approach all this the exact opposite way, so that a very small percentage of all adults go on clinical trials.
Over 70% of children
Yes.
are involved in clinical trials.
And it's considered wrong not to offer them studies. Flat out wrong. And that's the issue. And by the way, they've made advances even with old dumb drugs like chemotherapy, incrementally ratcheting up the cure. The presentation from Boston of the sequencing of a single patient's genetic material in their tumor to explain how they had become resistant to a BRAF inhibitor, one of the early drugs for melanoma. And this is amazing in itself, because this patient allowed for a sequential biopsy, a biopsy of tumor after treatment had failed, and when, frankly, his life was really quite brief, his life expectancy, and he knew it. And all he was able to say was, take this tumor and learn. And what was learned from him was the specific mutation that made his tumor resistant to a drug that had been working. And that will lead, of course, to potentially new drugs.
Yes, I'm really glad you brought this up, because even I have learned through this ASCO meeting, some of us, myself included, have early on sent our tissue for molecular profiling. But I'm learning that along the way, you may need to resubmit your tissue for new profiling, because as we learn more about the genome sequencing, it may be discovered that another pathway mutation, whatever it is, that can be identified and open the door for someone who may have an option of a different therapy.
The complexity can be overwhelming. I want to point several things out. New England Journal paper by the group in England a couple of months ago looking at renal cell carcinoma, and they sequenced one or several patients' tumors and metastatic sites. And they showed areas in the tumor that were consistently mutated, but they also showed tremendous heterogeneity in the tumors from the same patients. So that's the first challenge. When you do a biopsy and you think you know what you've been studying, have you sampled enough of the tumor to even be sure? Does the heterogeneity in 10% of the tumor ultimately end up being the problem, the part you missed? That's one thing. Two, does the tumor change, evolve over time? Some tumors seem to, others frankly not so much. For breast cancer, it's not been so clear. The third issue, which you touched on, is it is all well and good for us to sequence tumors. We are doing it, everybody's doing it. But we should not mistake the successful identification of a target when we have a drug for success. Success is not measured because I can give a drug that targets a specific mutation. Success is measured by the fact that when I give that drug, the outcome is meaningfully improved. And the truth is, we are going to identify many, many more actionable mutations and abnormalities than are effectively targeted. And I don't want to oversell this. So in this, there's a lot, because I've described time variation, geographic variation, and functional variation, all of which needs to be carefully sorted out.
How is a patient supposed to keep up on this, especially one with a very challenging disease?
Yeah. So first of all, let's not lose sight of some basics. This is not available to you unless you have consented to a biopsy, consented to the storage and testing of that tissue, and that means, by definition, that you're already in the hands of a team, usually, of physicians and others who are focused on this, which in turn means that you're likely in a center that is focused on research.
From the breast cancer perspective, what do you need from patients in the advocate community to help facilitate tissue collection?
Well, I agree with you, first of all, and we do it. We have in-house studies in my place where we do sequential biopsies over time in targeted populations to determine mechanisms of resistance to drugs. So let's not say for a moment that we're not motivated. But there are some barriers that are obvious. It can hurt to have a biopsy done. It can take some hours to go through a procedure. The tissue, as we spoke about earlier, may or may not reflect the totality of the disease you're studying. In that regard, it could mislead you. There's one other issue to point out, and that is that it costs money. And if it costs money, somebody has to pay. And so some insurers and others may not see that there's any value or benefit to this. Or they may say, with some degree of accuracy, that's research, and a research grant or research funding should be paying for that, not our covered premium payers. And so the financial aspect of this has to be noted. It also costs money to store that tissue, and especially to store it in a retrievable, annotated way so that when you are ready to test it, it's there, identifiable, and in the proper format, frozen, put in paraffin, all the things we have to do, DNA extracted. So I'm not disagreeing with you at all, quite the opposite. I spend a lot of my time doing this. But at the same time, it is regrettably not simply enough for anybody to say, I want to do this. You need a research protocol, you need standards, you need all this infrastructure in place because you don't want to do it and have it be for waste. And we have an obligation and responsibility to use it to ask good questions, not just the first thing that pops into our mind because it's expendable, that resource. I think that there are some answers. Certainly there's a potential for kind of liquid biopsies, blood tests that identify circulating bits of DNA from tumors, blood tests that identify circulating tumor cells or other reflections of the tumor that we can look at that would be less invasive, i.e. less expensive. So I think that the technology that's in development may make this question less challenging in the years to come. We are deeply appreciative of all patients who agree to undergo what look like medically unnecessary biopsies. That said, under certain conditions, it's possible that multiple biopsies will turn out to be a requirement for good treatment as we match specific changes and drugs.
Yes. Yes. So, in closing, as President-elect of ASCO, what's your dream and vision? What would you like, where would you like to be by the time your year is, your 12 months come to an end? What are your greatest goals and what you would like to focus on within yourself for your new role?
Well, I mean, ASCO is a big organization with many, many goals. Our mission, and I share it wholeheartedly, is a world without worry from cancer. And I want to make a point about that, because you got to listen to that very carefully. We're not imagining in a magical way that there's a world without cancer, but a world where cancer doesn't cause the degree of worry and anxiety is what we can, I think, reasonably hope for. And in my year, I hope that we can advance ourselves a little further along. It's worth pointing out that my presidential year will be the 50th meeting, the 50th annual meeting for ASCO, its 50th anniversary. And so we will have certainly a temptation to celebrate great progress, but I hope that we will really be forward-looking in terms of our promise yet to be delivered.
Thank you. Dr. Clifford Hudis, Chief Breast Cancer Medicine, Memorial Sloan-Kettering Cancer Center in New York City, and President-elect 2014.
Thank you very much.
Thank you very much.